A student cut a piece of onion and placed it on a microscope slide. The student then placed this slide on the stage of a light microscope and looked through the eyepiece. No cells could be seen in the piece of onion. Notice how they say a piece of onion, not a thin slice. Explain 2 ways, so that means tell me what they can do and then give a reason for why they should do this, in which this method could be improved to see details of the onion cells. So quite a lot of marks for this. So you basically have to talk about 2 reasons. One, state what you will do, 1 mark, then the reason why you will do it for another mark, and then you repeat that here. So look at this. This is onion cells under a microscope, and you can see them very clearly. You can see the nucleus, you can see the cell wall here. And the reason you can see this very clearly is because it's a very thin layer, so light can easily pass through. The other reason you can see it is you'll notice they're purple or blue, and they have a stain. This is called methylene blue, and this stain will stain DNA, so you can see the nucleus very clearly. But there are other stains as well, like iodine, which will stain the cell wall, so you can see that very clearly as well. So here are 2 ways in which you can improve this. There are many other ways. You can talk about changing the magnification, but I've gone for 2 really obvious ones. Use a thinner layer so light passes through cells. Here's what you do, here's why you do it. And then the next one, use a stain to see parts of the cell. Figure 4 shows mitosis occurring in some plant cells, and whenever they show you a picture of mitosis, they always show you 2 of the 4 stages, and the 2 they always show are metaphase, when the chromosomes meet in a line, meet for metaphase, or anaphase, when the chromosomes are pulled apart, A for apart, anaphase for apart. The cell in figure 4 was taken from rapidly growing parts of the plant. Which part of a plant has rapidly dividing cells? So most people probably guess this because it's a small piece of knowledge you either pick up or you just completely forget, but I'll tell you what it is anyway. So in a plant, you basically have regions basically at the tip of the plant, or where a stem sort of shoots off, or where it's sort of you get branching off from the stem, or even the tip of the roots down here. So you get basically here, 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 and in these regions you have rapid levels of cell division. So there's intense cellular activity in these regions. These are called meristems, and it's these regions where the plant grows. So the answer is meristem. Which stage of mitosis is shown in cell R? So that's this one, or that would be metaphase, because they're meeting in the middle, not being pulled to the either side, anaphase there. Bit of a tricky question this. Describe 2 genetic similarities of the new cells that would be produced in cell Q. So remember, in mitosis, what you're having is a parent cell, which basically swells and then divides to form 2 identical daughter cells. So in what way are these cells genetically similar? So there's no point saying, for example, they're the same size, because that's not a genetic similarity. Well, one, they'll contain exactly the same DNA. They're genetically identical. The second thing is they'll contain the same number of chromosomes as the parent. So the parent cell will contain 46 in a human, and this one will contain 46, assuming we're looking at a human here. So same DNA or chromosomes. You can either choose DNA or chromosomes, either's fine. And then diploid. Diploid is our word for saying the same number of chromosomes will be there, so 46. Haploid for sex cell, diploid for a body cell. So mitosis is producing body cells, so the cells will be diploid. These are the only 2 genetic similarities you'll need to learn. Di means 2, by the way, and ploid is sets of chromosomes. So a body cell has 2 sets of chromosomes in a human. 1 set from the mother, 1 set you inherit from your father. The cells in figure 4 were heated in hydrochloric acid. State 2 safety precautions that should be taken when heating hydrochloric acid. This is a really easy question. You just have to use common sense. You know that acid is corrosive, so you want to avoid contact with your skin and eyes. So 2 obvious things you should say: wear goggles and avoid direct contact, or wear gloves. Remember, state questions are the easiest. You just have to list something. You don't have to explain anything or describe anything complex. You just list. Another explain question. Explain one advantage of using an electron microscope to observe plant cells. So electron microscopes allow you to see things in much higher resolution, much higher magnification, so you can see things in much finer detail than a light microscope. So you can see this compared to something like this. Anyway, so explain one advantage of using electron microscope. So you could say either higher resolution or higher magnification for one mark. That's stating the actual difference, and then you have to give the advantage for it, which is so more detail is seen. Remember, 2 marks, so 1 mark for stating the difference and the other mark for giving the reason for that difference, the advantage of that difference.